I'll be discussing neuronal tau protein aggregates as a therapeutic target for Alzheimer's disease. To briefly introduce you to this protein, tau has 6 predominant isoforms generated from a single gene via alternative splicing, and it is found primarily in the adult central nervous system, where it is responsible for the stabilization of microtubules, neurite outgrowth, and axonal transport, and its functions are primarily regulated via site-specific phosphorylation. When phosphorylation state of this protein becomes imbalanced, tau can also play a pathological role. Tauopathies are neurodegenerative disorders in which hyperphosphorylated tau proteins are found in paired helical filaments, forming neurofibrillary tangles, or NFTs, which have a decreased affinity for microtubule binding. One type of tauopathy, Alzheimer's disease, presents itself in the form of 2 hallmarks. The first is a buildup of amyloid plaques composed of amyloid beta peptides, and the second is NFTs. Alzheimer's is the 6th leading cause of death and a major source of healthcare expenses. As our population ages, it is increasing in prevalence disproportionately to any other kind of disease. Delaying the onset or slowing the progression of Alzheimer's by 5 years could save $100 billion per year by 2020 in Medicare and Medicaid costs. While there are currently 5 FDA-approved drugs on the market, they merely act to slow down the progression of the disease without treating the underlying disease itself. Drugs aimed at treating disease hallmarks have so far failed at varying stages of clinical testing. Many of these unsuccessful drugs target the beta amyloid plaques hallmark. One reason this target may not be yielding effective results is that the buildup of these plaques occurs well before symptoms of cognitive impairments, and by the time the drugs are taken by subject with Alzheimer's, plaques have been in the brain, causing damage for a significant amount of time. A strong correlation between progression of symptoms and tau aggregate quantity suggests a significant role NFTs play in neurodegeneration. Thus, tau pathology may prove to be a better target for AD drugs. Though there are several different facets to target within the realm of tau pathology, we will focus specifically on 2 different aspects of tau biology that are currently being investigated as places to prevent tau pathology. The first is preventing the neuron-to-neuron spreading of tau, and the second is restoring the stability of microtubules. Research by Chai et al. Showed that anti-tau antibodies could be used to target and decrease tau pathology, implicating this as a therapeutic option for AD. The experiment studied the effects of 2 anti-tau antibodies that selectively recognize pathological forms of tau administered via intraperitoneal injection into 2 different types of transgenic mice expressing human mutant tau. The results were very optimistic in that they showed a showing of tau pathogenesis which resulted in a slowing of disease progression, preservation of motor function, and a decrease in degenerations of axons. The mechanism of action by which these antibodies work is unknown, but a paper by Clavigera et al. Showed that the injection of extracts of the insoluble mutant tau aggregates were necessary and sufficient for the spread of tau pathology, suggesting that, in vivo, tau spreads via an extracellular intermediate. One scenario hypothesizing why this therapeutic treatment may work to reduce tau pathology is that the antibodies bind and capture extracellular pathogenic tau aggregates as they are propagating from one neuron to the next, and thus prevent the spread of these molecules. A major function of tau protein is the stabilization of microtubules, and thus tauopathies manifest in the form of phenotypic features such as decreased numbers of microtubules, impaired fast axonal transport, and neurodegeneration. It has been hypothesized that drugs stabilizing microtubules may offset the loss of tau function that occurs in Alzheimer's when tau aggregates into tangles. Zhang et al. Also used transgenic mice models of tauopathies to investigate this hypothesis. They showed that the microtubule stabilizing drug Paxseed reversed many phenotypic effects of the transgenic mice. Treated mice showed an increase in fast axonal transport, decreased axonal degeneration, an increased number of microtubules, and decreased motor impairment. While these studies targeting tau pathology as a therapeutic treatment for tauopathies like Alzheimer's have yielded promising results, there's still a long path ahead in the translation of these laboratory results into clinical success. The next generation of Alzheimer's therapy must come in the form of drugs that are able to actually treat the underlying disease, but only time and plenty of clinical testing will tell if targeting tau is a ticket to slowing or reversing this prominent disease.